Well, hello, and welcome to Bio 211, Anatomy and Physiology 1. This is for a section at Norwalk Community College. And we're looking today at a generalized cell and how we actually will build this generalized cell. And there are some basic characteristics we want to begin with, and that would be, of course, that all generalized cells have a plasma membrane, and that plasma membrane is a phospholipid bilayer, if you remember from your reading, and it's going to form that outer boundary, and it separates the cell's internal environment, I, and external environment, E. Okay, and it's a selective barrier. And what we can do is we can limit what can move in and what can move out, arrows on these things so that makes sense, into a cell by creating these, and you'll see how we do that. It also can play a role in cellular communication, and from your reading you learned that you could put things on the surface of the cell, integral membrane proteins, and they can have molecules that will bind to them, and those molecules will then signal into the cell. Okay? But here's the plasma membrane. This is generally what it's doing. Okay? We think about the cytoplasm of a cell. So again, I'll draw a cell. And I'll keep some of the stuff there. There is that sort of receptor-like structure. And inside here is all of the cellulose. The cytoplasm is all of the contents that are found inside here. Okay? Where you actually have the cytosol, which is the fluid portion, which is mostly water, but remember that's also salt water, right? And it has specific organelles inside of it, and that could be, say, the nucleus over here, or some other organelle over here. But the point is that it's inside, it's suspended inside the cytosol portion of all of this, okay? And then finally, you have the nucleus itself. So even though it's part of an organelle, it's a large organelle that contains the DNA. And that DNA, even though your textbook says it's chromosomes, you should think of it as chromatin. If it's functional, okay, and consists of a single large molecule of DNA and the proteins that are required for it to function. So if you've read chapter 3, you know that we're going to require RNA polymerase to transcribe a specific gene. Well, that's the protein they're talking about here. And on every set of chromosomes, because there are 23 pair of these, they contain thousands of genes, okay, and those heredity units are going to give rise to what you see are the structural phenotypes of any organism, like look in the mirror. All of those physical structures that you're seeing are the result of the genes that were transcribed from those 23 pair of chromosomes, some of which came from mom and some of which came from dad. Okay? Cool. And this is a really pretty picture. Not realistic. You're not going to find this anywhere in Mother Nature. But it does the job of explaining to you, or at least suggesting to you, the number of things you could find inside of a cell. And I could just sit here and go through them and list them all off for you. I'm kind of hoping that you're going to take some time. You will draw these things out. Instead, what I'm going to do is I'm going to walk us through, from the material inside the text, what each of these things is and a suggestion of what they're doing. Okay? So let's take a look at that plasma membrane. That plasma membrane creates what we call a fluid mosaic model. And that fluid mosaic model has a sea of lipids, and those lipids are, of course, phospholipids, right? Phospholipids. Remember from AMP, from chapter 2, we're going to create a lipid bilayer, and that lipid bilayer is going to have within it either proteins or other lipid materials that are going to change the functionality of that plasma membrane, because we need to make sure that it is a regulated selective barrier. Okay? And that means we have to be able to let stuff in. We have to be able to let stuff out. Now we need to learn how we let it in and how we let it out. Okay? Cool. That lipid bilayer, made up of phospholipids, cholesterol, and glycolipids. Within it, we will put integral membrane proteins that extend through. Integral proteins, integral membrane proteins, MEMBRANE. Proteins extend into or through. Transmembrane proteins are a subset of these. They actually go the entire lipid bilayer. And peripheral proteins will attach to the inner or the outer surface, but they don't extend all the way throughout. So they wouldn't pass between both sets of phospholipid bilayers. Okay? To see this, take a look at this cartoon here. Let's start with peripheral protein. That's down here. It's not even actually in the actual membrane itself. A better example. There isn't a better example on here, unless I took something and stuck it inside the membrane there. Okay, but let's take a look at the blue structures. These are our hydrophilic polar heads, fatty acid, nonpolar tails, assembled tail to tail, but let's look inside here. We actually use cholesterol, and notice its structure, four cyclical carbon-based structure, and it's in between the phospholipid tails, and this increases the fluidity of this fluid mosaic model. This sort of helical structure here is the result of the construction of the amino acids that are found inside that specific protein, right? And in this case, it's a glycoprotein because it's a protein that is a integral membrane protein that actually has sugars that have been attached to it. And that's why we call it a glycoprotein. So it's been glycosylated. Okay, and if you don't get that, it's not the end of the world. What you want to see here is that we've added sugars to it. And notice, if you're looking on this side and you're looking on this side, what don't you see? The asymmetric distribution of sugars, glycosylations, are to the outer portion of the membrane here. Okay. Also, you're going to see that just because we have this one structure traveling through, it's not actually carrying anything. So the protein is just traveling through. Whereas if we create a channel, and notice here's a channel here, materials can pass now through that. Likewise, over here it doesn't look like we're moving anything, and we probably aren't. Instead, it's probably conveying a signal from the outside of the cell to the inside of the cell. Okay. So what are glycoproteins? These are membrane proteins of the carbohydrate group that are attached to it. That's all it is. And the glycocalyx is that sugary coating surrounding the membrane. And the purpose of this glycocalyx, of course, believe it or not, is to bring water to the cell. Bring water to the cell. And that's because, if you think about it, all those sugar molecules are attracting water. And if the water is moving towards the cell now, each and every single cell has the ability to pick up some water that's going to move into it. Those ion channels are going to be used, we're going to create ion channels, because not all integral proteins are ion channels. A subset of them are. And they can move these substances through the membrane. And the key word here, of course, is selectively. And what I should also put in here is with precision. And that's because those transporters are going to transport one ion type and one type only, unless it's a specific type of transporter that will transport two types of ions. And they may transport it in the same direction or in opposite directions. Receptors are for cellular recognition. A ligand is a molecule that binds to the receptor, and that will then cause a signal to enter into the cell. And then we actually will put enzymes at the membrane protein as well. That will catalyze chemical reactions either at the surface on the outside or at the surface on the inside. Okay, what is membrane permeability? Well, if we're thinking about how a membrane is built, right? Here is one phospholipid layer, here's the other phospholipid layer, and remember we have some cholesterol inside here, so just put C inside there, which means these things are all moving around, right? And every time they're moving around, there's going to be a little bit of space. And that means, say, O2, which is oxygen, right, can pass through. No big deal. Even CO2, let's do this. That's a C, believe it or not. O2. That can pass through as well. Things you probably don't want to have pass through, like carbon monoxide, can pass through as well. Okay? So it's permeable to some things. And because that steroid is there, you'll notice it decides to stay where things are at. And that makes sense, right? Because like dissolves like, and it doesn't want to be out here inside the water, so it stays inside that nonpolar environment. It is impermeable to say something like glucose. So let's just put a big G up here. Big G for glucose, which means glucose can't get in, which means we have to make something that's going to allow it to pass in. That would be that transmembrane protein. Now, for us to move things, it can be a passive process. So those gases passed without any extra energy required for them to move through. For us to move an ion through some of these channels, we might actually have to utilize an active process, right? Where we're going to use the energy from ATP to move something across the membrane. And usually, as I said a moment ago, against its concentration gradient, because that's where the work is. If you're moving something downhill, you don't necessarily need to expend energy on it. Okay. So one such passive example that is pretty much the basic mechanism for all life, right, is diffusion. And diffusion requires that there be a steepness of concentration. And that concentration gradient can be changed as a result of changing temperature. So if you increase the temperature, let's go increase, right? We increase the temperature, well, then the molecules are moving faster, and therefore they can move farther apart from one another. The mass, of course, if it's really large, let's say it's large, okay, it's going to move more slowly, right? Because it's too big. It's going to require more energy to actually move it around, okay? And then imagine, if you will, the surface area that it's trying to diffuse through, okay? All of these things will change the rate at which something is diffusing. And everything wants to get to what we're going to call as equilibrium. Now, I teach you that you don't want to be at equilibrium, right? And you'll see what I mean by that in general. So what's going to happen is, to be alive, what you're doing is we're letting diffusion help move materials, and then we expend energy to ensure no equilibrium. And hopefully that's clear, because if you're at equilibrium, you're at death, right? That's not to say that, we wouldn't want in a chemistry lab for things to be at equilibrium, but we're not a chemistry lab. We're a living organism, okay? That makes sense, I hope? Okay. I'm not sure what else I want to say about that. Nothing. So let's move. Okay. So there's simple diffusion, channel-mediated facilitated diffusion, and carrier-mediated facilitated diffusion. These are all examples of things that can diffuse, right? Look, oxygen molecule simply diffuses. We carry something through a channel, so it moves through. Glucose here in purple binds to a channel that's going to bind only glucose, and when that happens, when glucose binds this structure, it actually moves now into the cell. So the concept isn't difficult. However, you'll notice none of these things have been ionized. Now we actually have to do something where we're going to move and regulate, say, a charged molecule. And this, in this case, is going to be potassium. It's charged. And in some cases, it can move freely through when the channel pore is open, channel protein is open. But it can't when it's closed, which means we can create a gradient or regulate a gradient at the membrane, depending on which channels are open and which channels are not. You want to keep that inside the back of your mind. We can also move things down the glucose gradient, but limit the total amount that's inside of it. So you can have a specific concentration of glucose inside the plasma of your body. The interstitial fluid outside here has one concentration, and we keep a unique concentration of it here inside of the cytosol of the cell, because we don't want too much of it inside of your body. And we also want a certain amount of it here inside the interstitial fluid. We also need to remember that anywhere a solute will travel, the solvent will follow. And the solvent for everything inside of our body, primarily, shouldn't say everything, okay, is water. The net movement of water through a selectively permeable membrane from an area of high concentration to an area of low concentration is going to increase the total amount of water inside of that space, right? And water can pass through the plasma membrane in two ways. Through a simple diffusion mechanism through the membrane itself, which is really slow, right? Or through a series of proteins that are integral membrane proteins that I mentioned to you already. These are called aquaporins. We see many versions of these in the course of AMP1 and AMP2. Generally, though, we only talk about two of them, aquaporin 1 and aquaporin 2. Okay. So what happens when this water moves? Why is it so important? Because water is allowed to move wherever it wants to move. We've got some real problems. Here is a U-tube. Okay, and that U-tube has a left arm and a right arm, and it has a selectively permeable membrane inside of it. And at starting conditions, what's going to happen is we are going to watch as we have a certain concentration of solutes on the right arm of that selectively permeable membrane. And look what happens when equilibrium is made. Now, it's not equilibrium to the water. It's equilibrium to the mixture of water and the solutes. So now imagine this is a cell inside of your body, and we let glucose inside of it, right? So we put too much glucose inside there. What's going to happen? All that water is going to want to go in there. What's going to happen to the size of our cells? So what we do, okay, is there's a certain amount of force that's required to maintain this equilibrium. So what we end up doing is we expend a lot of effort that we call osmotic pressure to push that volume back. This is actually how you maintain equal volumes inside of your cells. You actually push materials in and out of a cell to make sure that the cell doesn't get to be too big, but it requires a certain amount of work, okay? So hydrostatic pressure will result in the amount of osmotic pressure required to get back to normal, meaning the cell itself is normal. To see this inside lab and here, we will do a take-home assignment for the lab, but the tonicity and its effects on red blood cells are easily seen here. Everybody remembers the names and what happens, right? But in an isotonic solution, that's where everything is normal. Things are moving between, and you maintain the normal shape. Hypotonic solution, you have the concentration of salts higher inside the red blood cell than they are outside the container, so water moves inside here. The opposite is for hypertonic solutions. You have very little salts outside here, so all the salt, you have very high salt concentration outside here, so all the salt, all the water exits to actually cause the cell to crenate. Okay? And this is what I mean by Mother Nature will expend energy to make sure that the interstitial fluid of your body is a specific tonicity. So you're a bag of salt water, right? Now, hopefully look at this cartoon a little bit differently. If that bag of salt water is too hypotonic, what happens to all the cells in your body? If the bag of salt water is too salty, what happens to all the cells in your body? This is why we want to understand why we have all of these ranges for all of the ions and materials inside of your body. And this is what we mean when we say you have a specific range you need to find these in. Got it? Cool. So let's take a pause here, and we will come back thinking about how active transport actually works.